So welcome, my dear friends. And once again, I'm happy to discuss some topic that can be useful for a student or for prospective student. A student can be a postgraduate or PhD student. Okay. So let us discuss, and the whole interest of the discussion is to keep aware about the different activities of the lab. And if you look at here, this Cancer and Translational Research Center is one of the lab led by me, led by Professor Nilesh Sharma at Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Dr. D.Y. Patil Vidyapeeth, Pune. And this is some additional information on Cancer and Translational Research Center. And here is just for the awareness and introduction to the prospective student, that can be PG or PhD student. Those are looking for their career or their journey in Cancer and Translational Research Lab. So, my dear friend, this is the building of the institute. This is the lab, Cancer and Translational Research Lab, where you can see the undergraduate student, PhD student, and as a team, we discuss every day for the betterment of research and science. For prospective student, these are also the QR code of different research IDs of Professor Nilesh Kumar Sharma, that is myself, and like Orchid, Scopus, with one ID, Twitter, to make a better connect with the prospective students, and those are looking ahead. And you can go through this website by scanning through the Google Lens. Okay, fine. So these are the introduction about the institute, laboratory, and the principal investigator or the professor, Nilesh Kumar Sharma. Okay. To also brief and introduce about the lab, if you look at currently the students, multiple students are making their journey in the laboratory, including undergraduate students, predominantly, postgraduate students, some summer intern students, okay, and PhD students. I think more than 25 undergrad, postgraduate, and the PhD student, they have been trained within the lab. And in the last 5 years, we have received a project from DST and also one project from ICMR DHR. Also, we have received DPU grant of very substantial amount as a seed grant, around 28.5 lakhs. The CTRL lab in the last 5 years is productive, and definitely the credit goes to the students, undergrad, postgraduate, and PhD student. So approximately more than 80 indexed publications are credited to the laboratory. Around 8 Indian patents, 1 is granted, and 7 are published. And now, so these were the introduction about the lab and for the prospective student. So Now it is important to discuss some of the trust area and the research project. The first research project I would like to describe here, that is of metabiopsy for the early diagnosis of cancer and inborn error of metabolism. And to achieve this approach, that is of metabiopsy, we have developed an in-house tool, that is of VTG tool, and I will explain later on. And if you look at here, the whole concept of metabiopsy, we have attempted to extend in the form of nail metabiopsy, urine metabiopsy, serum metabiopsy, sweat metabiopsy, tooth metabiopsy, saliva metabiopsy, and also the metabiopsy concept we have extended to the cellular and tissue metabiopsy. And in fact, this metabiopsy as a terminology is made up of two words, that is metab and OPSY. OPSY is a Greek term that defines the medical examination or procedures for the diagnosis of patients. Okay, and metab is derived from the word metabolites. Okay, so the metabiopsy is nothing; it is a innovative approach that helps to early detection of certain diseases, including cancer and inborn error of metabolic disorders. Now, it is, yeah, so here is the tool we have designed, fabricated in-house. So if you look at the GIF, that is the working model of VTG tool, that's what we call, and this tool has assisted us for the concept of metabiopsy. And left panel, if you look at here, this is the flow model of the VTG tool, detailed model, where how and what basis the metabiopsy is facilitated for the diagnosis of certain diseases like cancer and IMD. And there are a substantial number of publications are also we have produced by using this method and the tool, that is the VTG tool. And definitely, if you look at here, we have also considered and explored and evaluated our own metabiopsy tool in terms of very low. If you look at here, that is cost of VTG equipment. That is our own equipment. Operational expenses are very quite significantly low. It is simplest, least expensive, and unique design. And that is why we feel that VTG assisted metabiopsy can be a good option. Besides existing technologies in the field of early diagnosis of cancer and other disease conditions. Besides the metabiopsy, our lab, we also pursue the certain molecular aspect of metabolic and epigenetic pathway, how it makes a crosstalk. That means metabo-epigenetic axis in case of cancer patient by using cell culture model and also the tumor tissues. We also explore the idea of metabolite mimetics as anti-cancer drug. These mimetic we propose that can serve as a small molecule inhibitors of certain proteins or enzyme, that is pro-tumor or pro-cancer. Our lab also interested to explore certain other interesting aspect of cancer biology, that is xenotumor heterogeneity. And in this direction, we try to explore the certain anti-cancer role of dietary chemicals and the gut metabolized products. And that is also very exciting and challenging work we pursue in our lab. And the last project in the recent past, we have also conducted, that is the effect of anti-cancer composition as CUDF. If you look at here, GUDF. We also explore the DNA repair response pathway in case of anti-cancer drug like doxorubicin and cisplatin by using DNA repair inhibitors. We also explored in the recent past, and we conduct the role of small RNAs or microRNAs across the kingdom, like small RNAs from certain plant like maize and corn, how they can cross the barrier of human cell or cancer cell. So we also explore these type of the work. Okay. So the prospective student, like postgraduate and PhD student, those are interested. I would like to offer here the four major domain. One is the metabiopsy, early diagnosis. Other is the molecular understanding of metabo-epigenetic pathways. Third is the DNA repair response and certain inhibitors. Fourth is the metabolite mimetic as potential anti-cancer drug. And the last one, that is of small RNAs, research work as a anti-cancer from plant system to the animal system. And definitely the lab has grown up in the last 7 to 8 years, and we have huge number of our collaborators. That is Professor J.K. Paul, Professor Strode, Professor Gore, and many more. Professor Sinha, Professor Podar, and here is the lab members. And recently we have received some award and prizes. So these are the representative, the student participation, and they received certain prizes. I believe that this the information or what I have shared today can be useful for those students. Those are highly committed, dedicated, and sincere, and they are interested to explore their journey in the field of cancer and related metabolic diseases. Thank you very much for listening, and I hope that your journey will be brighter and prospective. Thank you very much.